Good afternoon, good morning, good evening, depending on where you are in the world. Welcome to one of our webinars today. It's a pleasure to have you with us. I'm glad that you're able to sign in and we're ready to go. I must apologize for the few minutes of delay. So today we're going to be COVID-19 testing on behind the scenes. And just as I prepared for this presentation or this web, if you're going to do have a nice chocolate to eat, you know, you have to think about something as simple as the cocoa needs to go into the farm, the cocoa needs to come out of the farm, the cocoa needs to be transported to a factory, the cocoa needs to be processed, it needs to be weighed, the quality of the cocoa needs to be checked. And then after all of that has happened, and then it goes into a factory that actually takes this cocoa, processes it up, and then we have chocolate, and then the chocolate gets redistributed around the world. And once the chocolate gets redistributed around the world, then the people such as myself, and I'm not going to call any chocolate by its name just in case I'm promoting any brand, but then we can all enjoy the chocolate. But there is so much that goes behind the scenes before we can enjoy a good chocolate. And just the same as there is so much that goes behind the scenes before a good test can happen. And so that's what I want to do today. I want to look at what happens behind the scenes of a COVID-19 test. So again, welcome. My name is Kilian Songwe. I am the president of the Global Healthcare Public Foundation and also the chief scientific officer here. A Global Healthcare Public Foundation has been around for the last 18 years, doing very interesting work on the standards and qualities and in the area of public health safety. So just to be able to look at what is going on, hold. Stop sharing. I like the fact that we just had a slight IT challenge, but this is the beautiful thing about our new world today. So as I was talking, so this is what we're going to look at in this webinar today. I'm going to take you through the emergency use authorization. We're going to talk about transport. Again, remember I spoke about trying to have a nice chocolate. It has to leave the farms, go to industry before you get chocolate. We're going to talk about transport. Then we're going to talk about country acceptance, and then the public use of the different test kits, and then the results. What does a result actually mean? So of course, today we've become very familiar with the temperature gun. The temperature gun, before you get into any office building anymore, there's going to be a temperature gun, the emergency room. These are some of the pictures that we've become used to in today's world. So talking about manufacturers, there are well over 670 different manufacturers for the COVID test kits that are out there. 327 of them will do molecular assays. This is for viral testing. And 343 of them will do tests for serological testing. Now, I think it is very important at this point that I should mention that we would call the names of many manufacturers and different tests that are out in the market. But I just want to make a disclaimer at this point and say that we are not promoting, nor are we advertising for any manufacturer or for any kit that there is. This is a very scientific, we're trying to take a scientific presentation and make it very simple, but we're going to have to use the names of some of the manufacturers, such as you see here. So talking about emergency use authorization. Emergency use authorization is what a lot of the manufacturers are using today to be able to get test kits out into the market. And basically the emergency use authorization allows the commissioner of the FDA and CE to basically allow, whether it's for a test or for a disease that is out there, that could either cause chemical, biological, radiological, or nuclear hazard to the people to be treated. So a lot of our manufacturers are using what has commonly come to be known as EUAs to be able to produce test kits. Again, I put in the slide so that we can see what is happening in the in vitro diagnostics area. There are a number of manufacturers from Roche all the way down to CSON that are producing test kits that are going to be used. And the test kits are going to be used in highly complex facilities, specialty laboratories, or in our general hospital facilities. So talking about the emergency use authorization, as I mentioned earlier on, this is out of the US Food, Drug, and Cosmetic Act, which basically allows the commissioner to be able to approve the use of a diagnostics for the treatment or testing of anything that might cause, as I said earlier on, chemical, biological, radiological, or nuclear threats to the common people. And I show you this here as an example of an emergency use authorization letter that is issued. And I want to just highlight the fact it says, this emergency use authorization, EUA, is being issued in response to the need for in vitro diagnosis device in detecting antibodies for SARS-CoV-2 during the coronavirus disease 2019. So it's very specific about what it is going to do and why it is issued. And next to it, it has a duration over which time it is going to be used. So you
Yes.
cannot have an EUA that doesn't have an end period. So for all EUAs, one of the big things that the manufacturers are trying to get their hands around is the sensitivity of the test and all the specificity of the test. Basically, those two words have been thrown around as the testing keeps on going. And basically, with the sensitivity, we're just trying to find out how often are we going to get a true positive. With this, we want to find out how often are we going to get a true negative as negative. So talking about the manufacturers and how many of them, as I said, 670 about, and the new ones coming into the business every day. Once they manufactured their test kits, now they have to get out of the manufacturing shops and they have to go out into the world. So there is transport that is involved. So in this supply chain, there has been a great consortium that has come together, the World Health Organization, of course, the World Bank, the Global Fund, different UN departments, the private sector, the Clinton Health Access Initiative, the Bill and Melinda Gates, Médecins Sans Frontières. There's a bunch of groups that have come together to form a really big consortium that is actually trying to help the different blocks of nations to be able to get the test kits that they need. So as a part of the supply chain and the utility of the consortium, basically what the consortium is trying to do is they're trying to work with suppliers so that they can negotiate access to the test and then also negotiate cost of the test. But they're also trying to negotiate equitable allocation to low and medium income countries. So this is very important to be able to use the consortium to be able to get the test kits that are required in the different blocks. So looking at the supply chain, I throw in this graph, the slide here, because it talks about the block and the countries And then equitable access will come because you're using the consortium. And with the consortium, you have coordinated purchasing, so you know exactly what a region needs. With coordinated purchasing, then you can have coordinated procurement. And in turn, once the procurement comes to the country, then the country can now streamline the distribution, as the case might be for different countries. So just to look a little bit into detail what happens with the consortiums, within the consolidated demand, the ministries of finance of all these countries need to be able to make the resources available. We need to think about what are the essential equipments that are going to be needed to test for COVID. One of the ones that has been overly spoken of is the PPEs, but there is something as simple as oxygen. Oxygen needs to be available for people who end up on ventilators. Then there is equitable access. Equitable access also comes with allocation of principles through the different task force in the different countries. And from my right, going to the left, the suppliers as well have to think about the demand that is being posed on them. And with the consortium, there can be a leverage in between the demand and the supply side. So when you leave from what happens at the consortium, then you come down to what is going to happen in the country. The country has to have validated, submitted requests. So every institution in a country cannot be ordering. There has to be a very clear, validated way of putting the request through the consortium. And the coordinating of the purchasing so that we know how much is coming into each country and the supplies are committed to by each country as they come into the country. And then, of course, the streamlined distribution of the commodities as they arrive, either in the different blocks of countries or regions of countries or in a specific country. So the supply chain is very important. And this is a lot of the things that are happening behind the scenes, which is completely unknown to the lay and common man out on the street who just wants to get a test done. So in the world today, again, and I'm going to just repeat, we're not promoting or supporting any of the manufacturers or producers or the airlines that you see here, FedEx or DHL. But again, IATA states today that 82% of all the freight in the world is done between FedEx and DHL. And now when you think about the fact that those two airlines do close to 82% of all the freight, which is a lot of medical supplies, and you think about the fact that to date, going down to June 30th, 4.5 million flights have been canceled. 4.5 million flights being canceled has a huge impact on cargo. And as a result, it has a huge impact on shortages of the testing kits and their availabilities in different nations. Forget the fact that 4.5 million flights have been canceled. When you look at the cost to business, that's $314 billion is going to be lost because of COVID and the transportation industry. But I use FedEx here just as a small example to be able to appreciate what is actually going on behind the scenes. So FedEx has had to now create extensions for deliveries. So if test kits were going to come out from the APAC region, from Thailand or China or Japan, these test kits will normally have been a day or two to get to Angola, Botswana, Kenya, Malawi, or Zambia. But now FedEx is saying it's going to be a minimum of 7 days and could even go up to 15 days. What's the impact of that? You see, FedEx has, for example, decided to suspend their express money back guarantee because COVID has actually made it impossible for them to guarantee that they're going to be able to fly into a country and fly out of the country as they would have liked to. Again, all these things are happening behind the scene, and the common man, all they know is, I want to get a test done. But now if the test kit can't even get to your country or get to where you are, it makes it very difficult for the test to be done. So now in a good case scenario, the test kits have arrived in your country, wherever the country may be. Once they arrive in the country, there has to be country acceptance. So when we spoke about the consortium, there are certain things that have to happen with the consortium. Your country must have acceptance criteria. But there have to be very simple regulatory approvals in the country of manufacturing. So the manufacturer must have made some very stringent requirements by the Food and Drug Administration or by the World Health Organizations. Then there must be in-country approvals for the different test kits to be able to be accepted in country And once they're accepted in country, every country has its own approval, methodology, or protocol that they are going to use for testing the different test kits to be able to ensure that they can be used. And to remember, because as I said earlier on, almost 100% of the test kits that are used today are made under emergency use authorization, which therefore means they might react differently to different populations. So each country is strongly requested to actually have a protocol to validate the different tests as they come into the country before they roll them out for use in the country. So basically, this is what would happen. There'll be an evaluation process which basically validates the fact that that test actually meets its intended use, and that there is a verification process that says that, yeah, we're going to get the results that the manufacturer told us that we're going to get. So basically, that's what happens with the validation and verification of the different test kits when they arrive in country. But what is very important, again, happening behind the scenes, and this is many weeks, many days, and many nights of very hard work by our scientists. They have to be able to ensure that the sensitivity of every test kit when it comes into the country, it is good for use with the local population. So basically, they need to ensure that the sensitivity of the test, meaning a true positive for every positive, is as high as 99 or 100%. And the true specificity, which is the negative test for every patient, is equally as high as 90 or 100%. So these tests are really important. And these are things that are happening behind the scenes. And thinking about the fact that we have 670 manufacturers out there, and all of them have test kits that they're trying to sell to different countries. What does that mean? It means if you're going to buy a test kit in a country, you're going to have to spend weeks doing validation and verification on that test kit before you can actually roll it into for public use. So looking at this again, looking at the sensitivity, I use this as an example to be able to say, this is what any country would like to see, or any group of scientists would like to see when they roll a test out. They would like to see that the sensitivity of that test is at least 99%, or the specificity is 99%. And this is what the manufacturers of the different test kits are going to make available as they put their different products into the market. So this is really, that's nice to know. Once the test kits are in the country, who is eligible for testing? Not everybody is going to get tested. Some people don't want to be tested. Some people don't need to be tested. So talking about testing, why? What is the big goal behind testing for COVID-19? Again, we just want to be reminded that the reproduction number for COVID-19 is for every one infected person, there are 3 potential new infections. So you want to have a testing goal, because the testing goal basically stops transmission and prevents the spread of the virus. And then, of course, it slows down the transmission. If we can find out more people and we can follow up their contacts, then that certainly reduces the spread of the virus. So looking into testing, basically, there are only 2 kinds of tests that actually exist for COVID-19. The first one is a viral test. And the viral test typically performed on respiratory specimens. Either it's a nasal swab. We've seen enough pictures, and I'm most probably going to show you a picture of a swab going into somebody's nostril and back into the back of their head. Or it's going to be a throat swab. Basically, this tests for the virus and the RNA of the virus, or the virus protein. And the second type of test is the antibody testing or serological testing, which basically is the use of blood samples. So, talking about eligibility for testing, there are priorities of people who need to be tested. People who are already hospitalized, they've showed up with symptoms, absolutely need to be tested. And we're going to look at the different kinds of tests that are available. Healthcare workers working in high pandemic areas, they need to be tested. And then, of course, there is another very nice population out there, which is the immigrant and emigrant population, people who are on the high move. I know that there has been a lockdown on a lot of air travel, but there is still cargo that is moving, and there's still some other chartered flights that are moving, and we need to be able to test in this area. So there are 3 priority areas for testing. I've just mentioned a few. Under priority 2, there are persons who are 65 years of age and older and may also have some other comorbidities. They need to be tested. Healthcare facility workers and first responders, this need to be tested. However, because of the shortage of the test kit and because the manufacturers cannot make them at the speed at which we want to use them, individuals without symptoms are not a priority for testing. So, looking into all of this, we want to go down into testing. All of these things are happening, again, behind the scenes. Every lay person would show up, or they will want to call their local health facility and they will want to be tested. But there is so much that is happening behind the scenes before they can actually get tested. Again, as I said, there is the whole manufacturing process, there is the transport process, there's the supply chain process that has to happen. And then when you come down to it, now you have to do the testing. So just before we get into testing, there is all this vocabulary that you can get from our website. These are words that have become part of our lexicon now, whether it's sensitivity or it's specificity, or you're talking about enzymes, or you're talking about PCR. These are words that we're going to have to come to know. So talking about the sample collection, if you're going to get a valid test result, this is the part where everybody is involved in. You have the clinician, you have the medical doctors, the nurses are involved in collecting the sample, and the sample has to be collected by somebody who is competent and well-trained. And as I said, basically the sample could either be gotten from the nose, it could be gotten from the throat, it could be a serological sample, it could be blood, or it could be stool. So those are the known samples that could be used today for serological test, for COVID-19 testing. So just so that you can appreciate some of the technicalities again that are happening behind the scene, more often than not, when you have a sample collected, it might not be tested at the site of collection. And if the sample is not tested at the site of collection, it means it's going to have to be transported to a laboratory that has the competencies to be able to test for that sample. And if that is going to happen, then there has to be storage mechanisms before the shipment of the sample. And again, these are things that are unbeknown to the layman when they show up to get a test done. All they know is, oh, my blood was taken, oh, somebody put a swab into my nostril and they put a swab into my mouth, now I want my results. However, there is still a lot that is going to go on behind the scenes. There are simple things like internal controls need to be done to ensure the quality of the test that is being done. Between the laboratories that are doing the test, they need to be able to do some interlaboratory comparisons to ensure that there is good quality results that they're being produced and that the results are reliable. So basically, talking about the testing itself, there are generally 4 tests that could be done for screening, diagnosing, and monitoring of COVID-19. The first 2, as I have on my slide, the reverse transcriptase polymerase chain reaction and the loop-mediated isothermal amplification. Those 2 tests are basically the viral testing. And the next 2, the lateral flow and the enzyme-linked immunosorbent assay, these are serological tests. So these are the 4 common tests that are on the market today for testing for the SARS-CoV-2 coronavirus disease. So let's look at the reverse transcriptase polymerase chain reaction. This is the one that most people have heard about. It's commonly called as the PCR test, and it's more or less kind of sort of the gold standard for testing for the SARS-CoV-2 virus. Basically, this test takes anywhere between 3 to 6 hours. It's a 3 to 6 hour test. So on a regular basis, you will be asked to come back the next day for your results. Most probably there might need to be a repeat test, or anything could happen while testing is going on. So generally, after the sample has been collected, you would be advised to come back the next business day to get your results. So what happens behind the scenes once the sample has been collected for a PCR test? This is a test that is heavily inclined, is done under a lot of temperature and a number of temperature cycles. So basically, once the swab has been collected, whether it's from the nostril or from the throat, that swab is brought down to the lab. A number of things happen in the background. the RNA from the sample is now taken and copied into DNA at a very high temperature of 95 degrees centigrade. The PCR test goes through a number of cycles of temperature rise and temperature drop. It drops from 95 degrees centigrade to 55 degrees centigrade, allowing for the strands to be able to rebind to the DNA. And then that's a cooling phase for rebinding. And then it gets heated up again to 73 degrees, allowing for new strands of DNA to bind. So there are cycles of temperature rises and temperature drops. And basically, there are about 30 cycles of this temperature rising and temperature drops. And what happens with every one of them is that regardless of how small the viral particle was that was in the sample when it came, it gets doubled. And so by the time it goes through 30 cycles of temperature rises and temperature drops, you might end up with about 35 billion copies of the one virus that was actually found in your system. This is very efficient because the duplication of the RNA into the DNA, copying into the DNA, allows, regardless of how small the viral particle was when the sample was collected, it allows for the technicians to be able to say, yes, fluorescent marker here, and the fluorescent marker is beginning to become bright, which therefore means there is a viral particle, and therefore you are going to be considered as having tested positive for the coronavirus. So that would be the PCR. And then there is the loop-mediated isothermal amplification, commonly known as LAMP. LAMP uses the exact same technology like the PCR, except for the fact that the temperature of 60 to 65 degrees. The same theory happens, but in this case, instead of the fluorescent markers happening, you actually have a cloudiness of the sample. And this cloudiness of the sample is actually due to a magnesium phosphate that is formed. This cloudiness actually allows you to be able to say, because the cloudiness appeared, then the person is actually positive for SARS-CoV-2 or COVID-2 as we've come to know it today. Now, those are the two viral tests. Now going into the next two, which are the serological tests. This is a cartridge, and this has been used, it's been seen many times. A lot of the ladies out there who might have used this for the pregnancy test. It's the same principle. A drop of blood is taken and it's mixed with a diluent, and it's put on the spongy part of the cartridge. And then it's allowed to flow through the cartridge. Basically, it moves from where you see the red drop and it goes down through the cartridge. It's called the lateral flow. But most people today have come to call this the antibody test. But its proper name is the lateral flow test. It's very important to note that the lateral flow test, commonly known as the antibody test, does not respond to the actual presence of the virus. That's the big difference between the lateral flow test and the PCR or the LAMP test. So what happens with the lateral flow test? You have a cartridge, and generally the cartridge would have 3 sets of lines, and the 3 sets of lines represent the control line, a control line for the antibody IgG, and a control line for the antibody IgM. These 2 antibodies, the IgM usually coming up first, and the IgG coming up later after infection with the SARS-CoV-2. So upon testing with the lateral flow, what you're going to get is, if the person is positive for IgM, you would see a line. You would have 2 lines, basically. You'd have a control line, which says that the cartridge is actually responding the way to either for the IgG or for the IgM. This capturing along the test and control lines, which can be seen by the eye, therefore producing 1, 2, or 3 lines depending on whether you're using a cartridge that does either the IgM or the IgG or does both. And if the cartridge did both, for example, here I have a combined cartridge. So this is a cartridge that is doing both the IgM and the IgG antibody test. So basically, The IgM would show up within the first 5 days of infection. So if within the first 5 days of infection, if you had a serological test done and you were using a combo, you might end up with two lines showing up. You have the control line and you have the IgM line. However, if you were already infected and you were down into the 10th day and you use the combo serological lateral flow test, you would end up with 3 lines: a control line, a line for the IgG, which is 10 days down into your infection, and a line for the IgM. Again, you don't see any of this. This all happens again behind the scenes. You don't see the cartridge, you don't see the lines. All you know is a sample was collected from you. In this case, it is going to be blood was collected from you. And all of these things happen in the box, way out of your sight. Now, the fourth test methodology here is the enzyme-linked immunosorbent assay, commonly known as ELISA. This has been around for a very long time. And basically with the ELISA test, we're looking at the antibodies, antigens and enzymes. There is a mixture of all of this in a well. And there's a reaction that goes through the wall in a very nice, easy 7-step process, which I'm going to take you through. So in the first 2 steps, basically, your blood or blood serum is put into the well, and antibodies, the patient antibodies stick to the walls of the well. So basically what you have there in the first cuvette, you see in number 1, your blood is being put into the cuvette. In position number 2, patient antibodies, they stick to the walls because the walls have been made ready for that. Then when you go to step number 3, the laboratory has produced antibodies with enzymes that they add on. So this is kind of like a double sandwich that is being created. And when you go into step 4, there is special coloring, special colorless molecule is added to the well. The special So in step 5 and step 6, the addition of the second layer of antibodies with a colorless molecule goes into the well and then it sticks. Now you get a color change. Once you see the color change on your plate, that's a spectrophotometer reading which the naked eye could see, or you could use a machine to read the density or the intensity of the coloration. And then at this point, you would be able to say that the patient is positive for the SARS-CoV-2 virus. However, if you go through the first 6 steps and you end up with no coloration, then this is a negative result. So you could get a positive result at step 6 with coloration, or you could go on to step 7, where there will be no coloration and therefore a negative result. So just to recap on the test profiles that we just spoke about, the PCR and LAMP are both viral tests, and the time period for the PCR is anywhere between 3 to 6 hours. The samples come from the nose or the throat, and this is generally done in a lab. 96 samples could be done at once. Or same case with the LAMP test, it's viral. Difference here, the time is a little shorter, 2 to 3 hours. Again, laboratory based, or it could be the point of care. It doesn't have to be in a hospital, a high complex hospital to be done. And again, 96 samples could be tested at the same time. So you could have 96 patients have their samples run at the same time. However, what that means is for patient number 1 who came in 4 hours earlier and has to wait for patient number 96 to come up before the test are run, they might be waiting a little longer. On the 2 serological tests, the lateral flow test and the ELISA test, the lateral flow test could be anywhere between 15 to 20 minutes, and that is done 1 patient at a time. And the ELISA test, again, we could do 96 patients at a time, and it's going to take you anywhere between 1 to 3 hours. So people wonder, why do I have to wait? Why do I have to come back to get my results? Again, because there is a lot of things that are going on behind the scenes. There are test controls that need to be run. We need to ensure that the results that are going to be given are reliable. So controls need to be run. We need to ensure that once a test comes out and is positive, that it is repeated so that we are certain that when we tell somebody that this result is positive, we truly know that the result is positive. But again, I just want to remind you, Again, most of the test kits that are out there under emergency use authorization. It is very important to keep that in the back of your mind, because you could get a test done, same methodology, same manufacturer, and a few days down the line, a positive result today could be a negative result tomorrow. That again goes back to the fact that under the emergency use authorization, basically the reason why manufacturers get the EUA is because they need to do more testing to ensure that the sensitivity of their test is as robust as it could possibly be. So that, of course, allows for them to continue to do tests. However, they need to be able to put out something that can be able to help treat and diagnose people immediately. So results. We've gone through 4 different tests that are available and methodologies that could be used. So what does a result mean? The test has been done. You've waited 2 days to get your results. So if it is a viral test, the PCR test or the LAMP test, basically what you get is you get a positive result or a negative result. If it's positive on the viral test, it means that you do have a current COVID infection. However, on the other hand, if your result is negative, it means for the viral testing, you do not currently have a COVID infection. If it is an antibody test or a serological test, as it has come to be known, if your test result is positive, you likely have had in the past tense a COVID infection. Or if the result is negative, it means you've never had a COVID infection. Now, when both tests are done, which is highly recommended in most cases, when both the antibody test and the viral test are done, we now look at the results together. You have a viral positive result. You likely have had a COVID infection. So you currently have the infection and you're beginning to build immunity for that infection. When the viral test is positive and the antibody test is negative, most likely you do currently have an active COVID infection, but you're still very early into the infection. This might be day 1 or day 2, and so you've not yet begun to develop antibodies. Remember, earlier on we did say that the antibodies, the IgM antibody, will begin to show up anywhere between day 3, 4, and 5. And the IgG antibodies will show up from week 2 and 3. So you could easily have a viral positive result and a negative antibody result because the antibodies have not formed. However, if you have a viral negative result and an antibody positive result, it means you had in the past or you are now recovering from a COVID infection. And this could be one of the people who are out there who are asymptomatic. They got up one morning, they felt a little funny, but by day 2 and 3, they're feeling okay and they're moving on. And if they go for an antibody test, yes, the antibody would most probably show positive for the IgG, which is a long-term antibody in the body, and negative for the viral test. On the other hand, if you have a viral negative result and an antibody negative result, you certainly know at this point that you've never had the infection and therefore have no immunity to the SARS-CoV-2 or COVID-19 virus. I'm going to repeat this again. It is possible for tests to give a negative result that is incorrect. It is very possible for that to happen. And as I mentioned earlier on, what the manufacturers are striving to get is they're trying to get to 100% or 99.9999% sensitivity. This is very important. And as a result of the fact that the SARS-CoV-2 virus is new, that's why you end up with the emergency use authorization, allowing for the public to have something to use to test and therefore keep the population safeguarded, but also allowing for the manufacturers to continue to collect data to ensure the sensitivity and specificity of their test. So again, just the limitations that are out there with regards to SARS-CoV-2 testing is the emergency use authorization. There's a big issue of supply chain. There are lots of countries that are trying to get test kits and can't get test kits. Poor quality of specimen. This is a new virus. People are learning how to collect the samples in this case. And reporting results. So you've gone in, you've had a test done, now you're waiting for your results, and the results have to be reported. Laboratories, different countries have different guidelines. Laboratories should follow the national reporting guidelines. There are different task forces in different countries through which the reports must go through before they actually get to the suspect. All positives and negative cases should be reported to national authorities. And again, in the case-based reporting, results should please get out within 48 hours. As soon as there is a result, they should be distributed within 48 hours of identification. Then there is aggregated reporting. Aggregated reporting is important and it is useful because the different countries are using this for epidemiological studies. So basically, at the end of the week, you want to know how many new confirmed cases exist in the country. You want to know how many confirmed deaths have occurred. You want to know how many confirmed discharges have actually happened, or the number of people that have been tested. So that's aggregate reporting, which is also very useful. So now we've gone from the manufacturing of the test kit, we've got the shipping of the test kit, we've got the acceptance of the different kits in the different regions and countries, and then we've gone into the actual testing, all the things that happen behind the scene. And then, of course, we all remember one of our very famous recoveries, Boris Johnson leaving the hospital, and down to my bottom right, that's one of the oldest COVID positive patients leaving the hospital. So there's so many of these cases going on, and so testing is important. Stay safe. The temperature gun is becoming more familiar. We need to sanitize, wash our hands, wash your hands, wash your hands. Wear the mask, very, very important. Maintain our social distancing so that we can all continue to stay safe. And our acknowledgements to the World Health Organization, CDC, FINE, associates at the Global Healthcare Public Foundation. Thank you. You could please contact us. We could be reached email, phone, Facebook, on our website, and we'll be very glad to provide you with any more information that you may need. But at this time, I would pause and go and take a few questions. If anybody has a question, you could please go into the chat box and just type out your question, and I'll read it and try to respond. Or if you want to speak to us, you could please just unmute and or get permission to unmute, and we'll be very glad to have a discussion about what you've heard. But again, these are all the things that go on behind the scenes before you actually get a test at your local health facility, and before you actually get a result. There is so much that has to happen. And all of these things are very important to ensure the safety of our different nations. So I'm going to stop at this point, and we'll take some questions, comments, or anybody who wants to make a remark. I'll be very glad to listen. Either this was very straightforward, people could enjoy the analogy between the cocoa coming out of the farm and the cocoa going into the manufacturing and coming out as chocolates. I guess that was a good way to start this. But again, that's exactly all the things that happen with for you to be able to enjoy a good chocolate happens for you to be able to get a reliable and good test done as well. Any comments, questions? Okay. I see a number of people going into the chat box, so I bet they're trying to type something or ask a question. We have 10 more minutes to go. Question? Can I test positive during incubation? Yes, you could test positive if you use the PCR test. Yes, you would. You would test positive but however, if you used an antibody test, the antibody test is certainly going to show negative in the first 3 days because the IgM antibody will still be developing. The IgM antibody comes into being. It starts showing signs from day 3 through 5, but more reliably from day 5 and up. And the IgG antibody doesn't come until much later, most probably about 3 weeks down into infection. So during your incubation phase, you really want to do a viral test, because with the viral test, the PCR test, once you get the infection comes in, again, as I said, with the PCR test, it goes through a number of cycles of temperature rise and temperature drop. And basically what happens with every temperature cycle is that the one little strand of virus in your body gets duplicated over and over and over again, which allows for the equipment to be able to read the positiveness of or the presence of the virus in your system. So yes, you would test positive during your incubation phase if you did a viral test, but you wouldn't test positive with an antibody test or serological test. Which professionals are supposed to test, and which qualification should they bear? Testing for COVID is a science, is a medical science, and so you have to be in a clinical laboratory. And as I said earlier on when I showed you the emergency use diagnosis, you could either be in a high complex laboratory, this would be a reference lab, or you could also be in a moderate complex laboratory, this would be a hospital-based laboratory. So you would have to be a medical technologist, a scientist with the right competencies to be able to test for the virus. This is not a test that could be done by anybody and everybody. Well, there are point of care tests, like the cartridge test, the lateral flow test. However, even for those, you still need to be trained, you still need to be deemed competent. I'm sorry, there was a second question. I missed the second question. What is the chemistry of soap in hand washing? The chemistry of soap in hand washing, first of all, it has to be an antibacterial soap. And in the absence of an antibacterial soap, we would like for you to use running water and soap, and not a soap bar. If it's liquid soap, it's most preferable to use liquid soap over a soap bar, because with a soap bar, you could actually end up, you just dirty the soap bar and you leave it for the next person. With liquid soap, you actually, again, you have to wash the hands for at least 20 seconds. You could do the 1, 1,000, 2, 1,000, 3, 1,000, until you get to 20. And you would have efficiently counted in your hands, drop it on the tap knob so that you rinse off any virus you might have left on the knob when you were opening. And then you could now, with on the knob, you could now close the knob, and you will be sure that you've washed off your hands. It's very important to use soap. It's more important to try to get an antibacterial soap, or if you don't have water, good running water and soap, then in this case, we strongly recommend that you use a sanitizer with at least 60 to 80% alcohol involved in it. That will be very useful in trying to maintain and reduce potential of infection. Then, of course, again, they say you want to keep your hands away from your eyes, your nose, and your mouth. It's very, very important to do that. Okay, one more. For how long does the serological COVID-19 test remain positive after recovery? So the IgM, the antibody IgM is going to disappear most probably in the first 2 to 3 weeks at the max, but the IgG goes on for a very long time. So the IgG antibody basically shows that you've had the virus before. The one thing we do not know about the antibodies to SARS-CoV-2 today is that we do not know whether they provide immunity to a possible future infection. That's the one thing 12 months out to be able to understand COVID-19 and be able to know what is true and what is a possibility. However, if you've been tested positive before, you could almost count at 100% that your antibody test, especially antibody IgG, is going to be positive for a very long while. This is going to be a few years down the line. Yeah. Okay. We could take 2 more quick questions. This topic is very important. Is there a plan to have this educational program to all? That is the entire nation. More public sensitization is needed. This webinar could be found on our YouTube channels. It could be found on our website. We're very glad to make it available to companies or individuals who would like to have it. We do this, this is gratis. So we'll be very glad to share this. If you find this important, you could please reach out and contact. We would like to do something for your group or for your workplace or any situation that arises. We'll be very glad to work with you, and we could make something very tailored that could talk to you about the testing. It could talk to you about ensuring your business continuity. We could talk to you about the standards that are involved in ensuring that you have an effective and efficient workplace to ensure continuity of your business. Just reach out to us you could reach out to us by email, or you could give us a call, and we'll be very glad to get in touch with you. One more. How do you test for COVID-19 using a blood sample? Is it possible to get soft copies of your presentation? Well, the presentation is going to be on our website. You could certainly see it. It takes us at least 48 hours to be able to upload this onto the website. Then you could have it from our website. What was the first part of the question? So yes, you could get this. You could actually get this on our website, and I think I just showed that again to you. You could reach our website there. How do you test for COVID-19 using blood samples? Basically, the blood just gets drawn from you like any other blood sample would have been collected. So it's a needle prick. A needle goes into your arm, most preferably your arm, and the blood is collected. And this is going to fall under the serological testing. And there are literally 2 serological tests that could be done, the lateral flow and the ELISA test that could be done. The lateral flow, very quick results in 15 to 20 minutes. You could get result depending on the manufacturer. And with the ELISA test, because that's a well test, that could take you very easily another 1 to 3 hours to be able to get your, for the test to be done. So it's going to take you about 6 hours for you to be able to get your results. But that's going to be a serological test, which requires blood to be taken, as opposed to the viral test, which is done with a swab through your nostril or your throat. Okay, one last one. Can someone who has recovered from COVID have a reinfection, or is it once-off disease? Again, as I said, COVID is very new with us. COVID is barely 6 months. COVID was found in December of 2019. As a result, it's named COVID-19. So we're still very early. COVID has not gotten a chance to go through a full cycle. So in the next 12 to 18 months, we're going to be able to, we'll be in a much better place to be able to say definitively whether there could be a possible resurge. And that's why I said a few minutes ago that the fact that you have antibodies to the SARS-CoV-2 does not necessarily mean that you shall not be reinfected. That information, as of today, we don't have. The disease the virus has just been around barely 7 months, and we need at least 12 to 18 months to be able to properly understand it, to be able to say, yes, this is a virus that will reoccur, or no, it's not going to reoccur. But again, just practice your safety measures, and you can ensure that it wouldn't be you who would be infected anytime soon. At this point, I say I'll take one last one. Is there one more? I see, I'm being told that there is one last one. Okay. What do we have? Do you really think that locally made cloth masks are effective and safe? The most important thing to note around cloth masks, and it doesn't matter where they are made, that they must have at least 3 layers. There has been a lot of studies the MIT Massachusetts Institute of Technology and Standards. There've been some that were studied out of South Africa, Kenya, Zimbabwe. The most important thing is they have to have 3 layers. And that middle layer has to have a polythene effect so that it can actually filter very small particles as they come through. So I expect the homemade mask, if they're done properly, if they're made properly, and you actually inspect them to ensure that they have an outer layer to the public, they have a middle layer, a polythene middle layer, and they have an inner layer that protects you, which is right on your mouth and nostril, then it should work as effectively as they intended. So now the homemade mask, or the cloth mask as we want to call them, or face covers as they're properly called, they should be able to protect you from large particles. And it is not a guarantee that because you have a face mask, you can now stop doing social distancing. No, you still have to do social distancing even with a face mask. But I can guarantee that if the face masks are made properly, 3 layers, then they will certainly reduce your opportunities or the chances of being infected with the SARS-CoV-2 or COVID-2 virus. At this point, I say thank you so very much indeed for joining us today for this webinar. We look forward to seeing you next week, 3 p.m. Uganda time, for another one. Thank you. Have a wonderful week. See you soon.